What can I tell you about this technology? All right, okay, I can share the results. Hold on, in the 71 or actually in 71 of one of these devices just to test in my lab. So in my lab, I've been measuring the effects of energy, scalar energy, and solar energy, and intermittent energy on three things: water, human DNA, and my own body. That part's recent. So I developed this method called electrochemical impedance spectroscopy. That forget it. But it's a method of measuring the flow of energy between two electrodes. So if I had a water sample and I didn't use my pendulum and I used my equipment, I would uh, measure the energy that flows between the two electrons. So what do you put in the middle? Water, DNA, and I was able to measure the electrical properties of water and DNA. So I really want to study the electrical properties because the body is electrical, the body is electrochemical, and the whole scientific community and the pharmaceutical industry study the chemical side. They're not studying the electrical side. And that's kind of where it's all at, in my opinion, because energy precedes and underlies the biochemistry. So if you can measure the energy, if you can treat at the energy level like this does, yeah, shed aside. So, Leon, what's my training go? Where was I? I meant to training go. I like to have it. Oh, summer silence. And I, just joking. Abandoning the electrical energy in the body is very important. We know that the brain is all electrical, obviously. Neurons conduct electricity. But it turns out that molecules also conduct electricity, particularly DNA. DNA is not only a conductor, it's a semiconductor and a superconductor. It's all three. So what DNA actually, is a helix, but there's a central channel down the center of the helix. And they discovered that electrons flow down that central channel faster than the theory predicts. That makes it a superconductor. That makes it quantum. That makes it really interesting. So I append, showing effects of these kind of energies on human DNA, that's kind of sexy. So, yeah. But the reason I chose DNA, because that's the reason I just explained. The DNA is, and you could buy it, not in your local pharmacy, but like there are stores that actually sell human DNA from female placenta. They cool this placenta, they put it in a big stirring in the back of your chamber and stir it all up, and then they separate out the proteins and the fats and the sugars and the carbohydrate, and you end up with pure DNA. So you might say, well, that's pretty artificial. That's not what DNA looks like in the body. That's true. And that's what Bruce Lipton would always say. It's like, well, you're prepared to study the pure molecule. And I got, why not? The molecule is the molecule. It has this shape. It has a double helix, magic shape. And if you want to study the electrical properties of a single molecule, actually, you do want to get rid of all the other stuff. But then you got protein and DNA. Well, what are you measuring? It's more complicated, and the body is so complicated that one of the reasons I don't work with people, or had not worked with people for so many years, was because people vary all the time. You come into the lab to do an experiment. You had a fight with your spouse for that morning, and you're a wreck. You're emotionally, so as humans vary from day to day, they vary from minute to minute. The biofield they were talking about, So what kind of measurement can you take on a system like that that's changing all the time? But it's complicated. So I said, okay, it's too complicated. Let's work at the molecular level and the cellular level. So I did a lot of work measuring the electrical properties of DNA, and which turns out to be incredibly sensitive to energy, external energy from a person, or your own internal energy, which you can control with your mind to affect your molecules and your cells and your disease, if you have such a thing, whatever that means. So the method that I use is impedance spectroscopy, but I modified it. The standard method, for example, uses two electrodes. An electrode is typically a cylinder, a metal cylinder. You put it in the water. Well, I use flat Tesla coils. So each electrode is a flat coil that's wound, like that. And the one on the other side is when I'm in the opposite direction. So you've got counter wound electrodes. I use dissimilar metals, silver and gold, and that in itself creates all kinds of cool energy exchanges between the two electrodes. And then when I take my measurements of DNA, for example, the machine that I have allows me to take a measurement at the resonance frequency of the target. And this is important because if you want the sensitivity and the specificity, you can't just take a regular conductivity meter, stick it in the water, and say, there's my measurement, because it's the wrong frequency. It's measuring at some arbitrary frequency. An electrical engineer decided to make this machine, function at 2 kilohertz, and that's the wrong frequency for DNA, maybe for some things. So with those three modifications of the standard method, I developed a very sensitive method. And I actually tested the energy from this thing on the DNA, and yes, it does have not a super strong effect on the DNA, which is interesting. So it's not, DNA is so sensitive to virtually all energy that I can say that this energy is not specifically designed to resonate with DNA. It does a little bit like everything else. But when I moved on from measuring DNA to measuring cells in my own body, now we're talking really strong effects. So let me elaborate on that. So again, since the body's so complicated, let's use cells. The most accessible cell to get from the human body is blood cells. And I've actually just finished a study where we're playing music to blood cells. He did that, and I said, well, I don't want to work with blood, man. Yeah. I'm going to work with hemoglobin, which is one of the big molecules within blood. So anyway, let's use it. You're into music. Find the sound. Yeah, well, we haven't done that yet. That's another thing we could do. So I decided, and every time I do an experiment, I need a phlebotomist to take blood from it. I mean, come on. I think experimented a lot of the night. We've got to work on. Where's my local phlebotomist? Can you come over? I want to bring his guy. That's not going to work. So I started reading scientific literature and discovered that cheap cells are a valid, legitimate scientific way to get cells for the body. You take a knife in your mouth, scrape the inside of your cheek, transfer the cells to water, and then stick the electrodes in. And I started doing that recently, and it turns out that method is very sensitive. Again, maybe because of the way I modified the measurement method, or maybe because my cheek cells are particularly sensitive. But I've done it well again. And it turns out that cheap cells. So the method is so sensitive, for example, that I can measure the difference between drinking regular water and which causes, if I drink like a little under a liter of regular water, and then I wait about half an hour and I take my measurements, the conductivity in my cheap cells increases by about 5%. So I'm going, man, that's not much. So what I did was drank structured water, very similar to that structured water, because we both work with the same scientists, two scientists, and both got involved with structured water. When I drink structured water, the conductivity goes up by 45%. So that's a huge difference, and that shows you how sensitive that method is. Then I went and studied, what did I do with this thing? I sat in the room not that far away, because I use my pen away, but I'm dowsing a guy. Actually, the best place where the energy radiates the strongest is like here. But what am I going to do? Sit on it there? So that wasn't practical, and so that I was so I sat, had a distance, and then I used the voice program for my own frequency, and I just did like 2 cycles, which is like 9 minutes Above 24 minutes, 20 minutes, yeah, which is very small amount. This thing produced a 80% change in 24 minutes. So big difference, a huge effect. So that kind of blew me away, and that's partly why I'm here, aside from our residents, kind of why it's. I'm like, Matt, whatever she did, she's riding us up, because it really works, and it really works well. So that's what I can say about that. So